this video, I want to go over the root causes of insulin resistance, cardiovascular disease, and systemic inflammation. However, first I want to go over some of the basics of insulin resistance and diabetes. When we eat a meal, that glucose from our meal is absorbed into our bloodstream. And it's important to realize is that certain cells, like liver cells and brain cells, can immediately take up that glucose and use those glucose molecules as a source of energy. However, other cells like skeletal muscle and adipocytes, they need help to take up that glucose. They're not able to immediately take up that glucose. So how do skeletal muscles and adipocytes take up this glucose? What happens is the pancreas senses that glucose in the bloodstream. So in response, the pancreas releases insulin. And this insulin helps the skeletal muscle and adipocytes take up these glucose molecules. So how exactly does insulin help these tissues take up those glucose molecules? Well, what happens is that insulin binds to this insulin receptor. So once insulin binds to this insulin receptor, it activates a signal transduction cascade that leads to the expression of these glucose transporters on the plasma membranes of these cells. So now that we have these GLUT transporters on the plasma membranes of these cells, now they're able to take up those glucose molecules. And if you're interested in more details, essentially the insulin molecule binds to the insulin receptor. And then it activates the signal transduction cascade that leads to these vesicles with these GLUT transporters fusing with the plasma membrane, and now you have these GLUT transporters on the plasma membrane, so now the cell is able to take up the glucose molecules. So that's how insulin helps these tissues take up glucose molecules. They lead to the expression of these GLUT transporters that allow them to take up glucose molecules. So what's important to realize is that skeletal muscle and adipose tissue make up the vast majority of tissue in our body. So therefore, they're responsible for removing the vast majority of glucose from our bloodstream. However, this insulin is very important to allow these tissues to remove the glucose from the bloodstream. And what happens in type 2 diabetes are these tissues do not respond to insulin. When the insulin binds, this signal transduction cascade is inhibited, and therefore the cells are not responding to the insulin. So when these tissues do not respond to the insulin, then they will not take up the glucose molecules, so then the glucose is stuck in the blood and you have high blood glucose levels. So how exactly does this happen, and what does type 2 diabetes look like clinically? So let's say these graphs represent the time course of type 2 diabetes. So first, the skeletal muscle is going to become insulin resistant, where it's not responding to that insulin very well. So even though the insulin binds, it expresses very little GLUT4 transporters. So therefore, to compensate for the skeletal muscle insulin resistance, the pancreas is going to release excess insulin. So we're going to see high levels of insulin. So now that we have high levels of insulin, all these insulin molecules are going to apply a lot of pressure to force the skeletal muscle to respond, and therefore respond to that insulin, expressing those GLUT4 transporters. So now the skeletal muscle can take up those glucose molecules. So this is the beginning of type 2 diabetes. Again, first, skeletal muscle becomes insulin resistant. So to compensate, the pancreas releases excess insulin. So you're going to see high levels of insulin. And those high levels of insulin are going to keep glucose levels in check. So we're going to see normal glucose levels. However, eventually what's going to happen is the beta cells in the pancreas are going to become exhausted, and we're going to see beta cell dysfunction, and these beta cells are going to begin to die. So once these beta cells begin to die, now we're going to see much less insulin. So we're going to see the insulin levels decrease. However, remember, the skeletal muscle was already insulin resistant. So now that we have lower levels of insulin, and the skeletal muscle is resistant to the few molecules of insulin we do have around, this is going to lead to the skeletal muscle not responding to the insulin. So therefore, we're going to have very low levels of those GLUT4 transporters. So now all that glucose in the blood has nowhere to go. So we're going to see hyperglycemia. We're going to see those blood glucose levels rising. And we can see that here. So when we see hyperglycemia, that's a sign that you're very late in the diabetes progression. So again, this is what the progression of type 2 diabetes looks like. Again, first the skeletal muscle becomes insulin resistant. So in order to compensate for that skeletal muscle insulin resistance, we're going to see the insulin levels increasing. But eventually the beta cells in the pancreas are going to get exhausted and those beta cells are going to die. So then we're going to see the insulin levels drop. So now that the insulin levels drop and the skeletal muscle is still insulin resistant, that's going to lead to the blood glucose levels increasing. And then you have type 2 diabetes. And then you're going to have all the microvascular complications associated with hyperglycemia. But now you might wonder, why does the skeletal muscle become insulin resistant in the first place? So there's a lot of evidence that insulin resistance begins in the adipocytes. So let's say we have these two people, but let's say one person begins to develop hypertrophic adipocytes, where we have these adipocytes that are filled with excess triacylglycerides. This, through many different complicated mechanisms, is going to lead to the adipocytes releasing these inflammatory cytokines. For example, as the adipocytes become hypertrophic and they grow in size, the center is going to become hypoxic, where again we have those oxygen molecules diffusing into the cell, and the center of this adipocyte is going to have less oxygen because there's going to be less diffusion and less oxygen reaching the center of this adipocyte, which is going to activate THF-alpha, which is going to lead to a pathway that leads to the release of these inflammatory cytokines. Also, as these adipocytes grow in size, they're going to run into each other, and that's going to cause physical stress and physical forces, which will activate mechanical gated channels, which will also activate a signal transduction cascade that will lead to the release of these inflammatory cytokines. Also, this excess triacylglycerides lead to metabolic stress, which also releases these inflammatory cytokines. But the point is when you have adipocyte hypertrophy, that's going to lead to the release of inflammatory cytokines. And these inflammatory cytokines are going to attract macrophages and different immune cells, which are going to release even more inflammatory cytokines. So these hypertrophic adipocytes release inflammatory cytokines, which is going to bring macrophages, which are going to release even more inflammatory cytokines. So now we have a lot of inflammation. But something important to realize is we have these adipocytes all over our body. So this is going to lead to systemic inflammation. And these inflammatory cytokines are going to enter the bloodstream, so now we're going to have systemic inflammation. But what's really important to realize is once you have those hypertrophic adipocytes, that's going to lead to systemic inflammation and lipotoxicity, where there's excess free fatty acids in the bloodstream. And these are the two biochemical mechanisms that lead to insulin resistance in cells. And we'll talk about this biochemistry later. But so now you can see how hypertrophic adipocytes can lead to insulin resistance. But what's important to realize is that genes can play a huge role in this entire process. For example, some people have genes that give them what they call a high personal fat threshold, where they can have very hypertrophic adipocytes before they activate that entire process that we've spoken about. While there are other people with genes where they go through very minimal adipocyte hypertrophy, which will then activate that entire process, leading to systemic inflammation and lipotoxicity. However, eventually, slowly but surely, these hypertrophic adipocytes will eventually lead to systemic inflammation and lipotoxicity, which will lead to not just type 2 diabetes, but also the progression of atherosclerosis. So how exactly does systemic inflammation and lipotoxicity lead to atherosclerosis and type 2 diabetes? So now we have the adipocytes releasing free fatty acids and inflammatory cytokines into the bloodstream. So what's going to happen as a result? Well, keep in mind, we have immune cells like macrophages all over our body in all different tissues. So these macrophages are going to respond to those free fatty acids by releasing even more inflammatory cytokines. And if you're curious on the mechanism, these macrophages, they take up this saturated fatty acids, which alter the plasma membrane, which then activates this protein, which activates the inflammasome, leading to metabolic inflammation. So not only are the adipocytes releasing these inflammatory cytokines, but these free fatty acids that are released into the bloodstream essentially induces these macrophages all over our body to release even more inflammatory cytokines. So now you have massive systemic inflammation. But also these adipocytes are going through uncontrollable lipolysis, leading to the release of massive amounts of these free fatty acids. So that's going to lead to ectopic fat deposition, where we're going to see lipids depositing in tissues where they should not be. And this is also going to contribute to the formation of visceral fat, which is fat deposits in our viscera near our internal organs. And the truth is, the deposition of visceral fat is a very complex process, and many people think that cortisol is actually the primary factor leading to visceral fat formation. There are experiments that show that higher fructose leads to higher visceral fat formation, and also insulin. There are other studies suggesting that insulin leads to visceral fat formation. But we know that this lipolysis from these adipocytes can deposit, leading to visceral fat formation. So this visceral fat is extremely metabolically active, which means it's going to release a lot of free fatty acids, and it's going to release a lot of inflammatory cytokines. And what's important to realize is, due to the anatomy of the body and the way the anatomy is set up, all those free fatty acids and inflammatory cytokines that are being released from this visceral fat is going through the portal system, which is going straight to the liver. So therefore, this visceral fat is releasing a lot of free fatty acids and inflammatory cytokines to the liver, contributing to liver fat. So not only is this uncontrolled lipolysis from the subcutaneous tissue leading to lipotoxicity and therefore those free fatty acids going to the liver, but this visceral fat contributes to liver fat formation. And there are really neat studies that show when you transplant visceral fat into mice, that leads to fatty liver formation, which then leads to insulin resistance. And there's also really nice reviews by Marchesini and Schrimmer et al. That show that fatty liver is one of the best predictors for metabolic disease. And fatty liver correlates very well with other markers of metabolic syndrome and metabolic disease. So you might wonder, how do we develop a fatty liver, and why does fatty liver correlate so well with markers of metabolic disease? So there are many factors that contribute to hepatic lipogenesis. One major contribution is through this uncontrolled lipolysis, where these adipocytes go through lipolysis, releasing these free fatty acids that are taken up by the liver and are used to form these triglycerides. Another factor is these branched-chain amino acids. So branched-chain amino acids can induce hepatic lipogenesis through many mechanisms, but one major mechanism is through this particular pathway, where it leads to the formation of citrate, which is then used for this de novo lipogenesis, forming these triglycerides. Another major factor is fructose. So it's a little complicating, but the point is when fructose enters the hepatocyte, it goes through this unregulated pathway that forms a ton of acyl-CoA, which then forms a lipid droplet, and that leads to hepatic lipogenesis and the formation of these lipid droplets made out of these triacylglycerides. However, there's another important mechanism that fructose uses to stimulate hepatic lipogenesis. So when fructose gets metabolized by the liver, it forms this toxic methylglyoxal. So why is this methylglyoxal so toxic? Well, it inhibits AMPK, which then leads to the formation of fat and stimulates fat synthesis. However, constant inhibition of AMPK leads to serious long-term side effects. For example, when you do the opposite, when you take metformin, that activates this AMPK, and that leads to lower levels of diabetes and cancer and heart disease and neurodegenerative diseases. And I know this sounds too good to be true, but look it up for yourself. And they've done studies with literally looking at hundreds of thousands of people who take metformin, and it's shown that they have lower levels of diabetes, cancer, heart disease, and neurodegenerative diseases. So again, this metformin activates this AMPK, which leads to these healthy outcomes. But again, constant fructose essentially does the opposite. Constant fructose leads to methylglyoxal, which inhibits AMPK, which leads to a lot of negative adverse outcomes. For example, one of them being increased hepatic lipogenesis. And again, I know this sounds too good to be true, but really just look it up for yourself and come up with your own conclusion. So now the liver is left with all these triacylglycerides. But you might ask, why doesn't the liver just go through beta oxidation? Why doesn't it take these triacylglycerides and the free fatty acids and oxidize them through beta oxidation? Well, a key characteristic of insulin resistant in any tissue is mitochondria overload, where these mitochondria are essentially overloaded with substrate. This liver is accessing too much fructose and free fatty acids and branched chain amino acids in an unregulated fashion, where you're simply overwhelming the mitochondria. There's simply too much substrate, so it can't oxidize all these substrates, so that leads to excess formation of fat and triacylglycerides. And that's a key characteristic of any insulin-resistant tissue. So we know in the context of type 2 diabetes, we're going to have fatty liver and we're going to have systemic inflammation. So what are the consequences of fatty liver and systemic inflammation? Well, these are the two biochemical mechanisms that stimulate insulin resistance. So we can see through this diagram that these triacylglycerides and their acyl-CoAs will essentially lead to interacting with this JNK1 complex, which will then react with the insulin receptor substrate, leading to hepatic insulin resistance. And that systemic inflammation also contributes to hepatic insulin resistance. For example, inflammatory cytokines like interleukin-6 leads to the activation of the SOX-S3, which leads to hepatic insulin resistance. And the same thing with TNF-alpha that activates NF-kappa B, which also leads to hepatic insulin resistance. So that systemic inflammation from these inflammatory cells also contribute to these liver cells being insulin resistant. So what are some of the consequences of having a fatty liver that's insulin resistant? Well, remember, these adipocytes are going through uncontrolled lipolysis, releasing these free fatty acids that are taken by the liver. So the liver is going to take these free fatty acids to form these triacylglycerides. But this process is also going to induce the liver to secrete these VLDL particles. Then these VLDL particles are going to drop off free fatty acids to tissues like the adipocytes. So now the adipocytes regains those free fatty acids. And we know once it gains those free fatty acids, it's going through uncontrolled lipolysis. So it's going to release those free fatty acids, which are going to be taken up by the liver. So you can see how this is going to stimulate a vicious cycle of VLDL secretion and lipolysis, which will increase the levels of VLDL, which subsequently will increase the levels of LDL. But not just that, when these VLDL and LDL particles are dropping off free fatty acids to tissues, they're also going to drop off some of those free fatty acids to skeletal muscle. So this skeletal muscle is going to gain intramyocellular fat and lipid droplets, which is very important for skeletal muscle insulin resistance, which we'll talk about later. But remember, these hepatocytes are also insulin resistant. So that's going to lead to uncontrolled gluconeogenesis. Because remember, normally insulin inhibits gluconeogenesis. But because these hepatocytes are resistant to insulin, they're not going to listen to insulin, so they're going to go through uncontrolled gluconeogenesis, increasing the blood glucose levels. So that's going to lead to the pancreas responding and compensating by increasing the insulin levels. So what does this look like? So now the liver is going to go through uncontrolled gluconeogenesis, secreting a lot of glucose into the bloodstream. But fortunately, we have the pancreas to compensate for this excess glucose secretion by secreting excess insulin. And that insulin is going to keep those glucose levels in check, because we know that insulin is going to bind to the insulin receptor, stimulating the expression of these GLUT4 transporters, which then allows the skeletal muscle to take in those glucose molecules, keeping the blood glucose levels in check. So this is going to work fine, and we're going to have normal levels of blood glucose as long as the skeletal muscle is insulin sensitive. However, in this stage of diabetes, is the skeletal muscle going to be insulin sensitive? Well, remember, the adipocytes are going through uncontrolled lipolysis, releasing a lot of free fatty acids, and some of those free fatty acids are going to be taken up by the skeletal muscle. And not just that, remember, the adipocytes are going through lipolysis, releasing those free fatty acids that are taken by the liver, which then secretes those free fatty acids and triacylglycerides in the form of VLDL, which is also going to drop off free fatty acids to the skeletal muscle. So this skeletal muscle is going to have a lot of free fatty acids. And not just that, remember, we're going to have systemic inflammation. We already explained how those hypertrophic adipocytes release inflammatory cytokines. And we already explained through different mechanisms how the immune cells and macrophages also will release inflammatory cytokines. So we have a lot of inflammatory cytokines. And unfortunately, these are the two mechanisms that lead to skeletal muscle insulin resistant. For example, these free fatty acids, through different mechanisms, essentially form these lipid intermediates which lead to skeletal muscle insulin resistance, inhibiting this insulin pathway. And also those same two inflammatory cytokines, that IL-6 and TNF-alpha, also through different mechanisms, lead to insulin resistance in skeletal muscle. So this is the perfect storm for having insulin resistant skeletal muscle. So again, we're going through uncontrolled gluconeogenesis. So to compensate, the pancreas has to release a lot of insulin. And that insulin is going to tell the skeletal muscle to take up the glucose molecules. But because the skeletal muscle is insulin resistant, the pancreas is going to have to release excess insulin, so that's going to lead to hyperinsulinemia. However, if these beta cells in the pancreas are constantly releasing insulin, that's going to eventually lead to beta cell exhaustion and death of these beta cells. So then we're going to see a significant drop in the insulin levels. So if we have low levels of insulin and the skeletal muscle is resistant to the little bit of insulin we do have around, that's going to lead to hyperglycemia. And our blood glucose levels are going to go out of control. So you might wonder, why do we go through beta cell exhaustion and death of these beta cells? Well, nobody really knows, but some of the leading theories are glucotoxicity and lipotoxicity, where excess glucose levels and excess lipotoxicity with these high levels of these free fatty acids in the blood lead to beta cell death. And there are studies that show prolonged hyperglycemia leads to changes in beta cell specific transcription factors. And also, high free fatty acids in the context of hyperglycemia has also been shown to lower the expression of insulin. Another factor is essentially whenever the beta cells secrete insulin molecules, they also secrete this amylin compound. And this amylin compound essentially inhibits and leads to beta cell death. So it's kind of like a negative feedback circuit. Also, due to the way insulin is secreted and due to the secretory pathway, whenever insulin is secreted, that leads to ER stress, which activates the unfolded protein response, which can then lead to apoptosis and cell death of the beta cells. One last mechanism is essentially constant activation of mTOR. So we know if you're constantly eating meat and the amino acids associated with meat, and constantly eating a high glycemic load, stimulating the hormones associated with a high glycemic load, those are the two factors that stimulate mTOR. And if you're constantly stimulating mTOR, that's going to lead to beta cell proliferation and mitosis. And if you're constantly stimulating the division of these somatic beta cells, that will eventually lead to cellular senescence and theoretically beta cell apoptosis, leading to the death and the destruction of these beta cells. So the truth is, no one really knows the cause of beta cell exhaustion and whether it's reversible or not, so it's a highly debated topic. But you might wonder, who cares if you constantly have hyperglycemia, or if you temporarily have hyperinsulinemia, or uncontrolled lipolysis and systemic inflammation? Why should you care if you put your body through these conditions associated with type 2 diabetes? So what are some of the consequences of hyperglycemia? Well, these glucose molecules are able to open up and form this linear form of glucose. And this is a problem because these linear forms of glucose molecules can react with proteins. And it doesn't matter if it's a cytosolic or extracellular protein, the chemistry is the same, where these amine groups on the proteins nucleophilically attack these aldehyde groups of these linear forms of carbohydrates. So then when we do that, we go through this unregulated set of chemical reactions where first we form a Schiff base, then we form an amidori product, which then forms a 2,3-enediol. Now once we form this 2,3-enediol, it's going to react with oxygen. And when it reacts with oxygen, we form two important products. First, we form hydrogen peroxide. And this hydrogen peroxide is really important because it can open up and form this hydroxyl radical. And this hydroxyl radical is extremely toxic. It's a reactive oxygen species, so it's going to react with, modify, and damage biomolecules like DNA. And this can cause DNA mutations, which can contribute to cancer. But not just that, we also form these advanced glycated end products, which are essentially just proteins that have been glycosylated in an unregulated fashion. So therefore, excess glucose reacts with proteins in an unregulated fashion to form these advanced glycated end products. So hyperglycemia can lead to the formation of these advanced glycated end products. But what's important to realize is that the fructose molecule is 7 to 20 times more likely to form these advanced glycated end products than glucose molecules. But once we form these advanced glycated end products, they cause lots of problems. For example, they increase vascular permeability, they increase the amount of oxidized LDL particles, and they also lead to inflammation. So how do they do this? Well, essentially we have immune cells with these RAGE receptors that sense these advanced glycated end products. And once these immune cells sense these advanced glycated end products, that leads to the formation of these inflammatory cytokines. And these inflammatory cytokines induce inflammation, which leads to the production of these reactive oxygen species, which we've already explained can lead to DNA mutations, which can potentially lead to cancer. So insulin resistance can lead to hyperglycemia and systemic inflammation. But not just that, later on we'll see how insulin resistance can increase LDL levels. So this leads to the perfect storm for atherosclerosis. So how does this happen? Well, LDL in an environment of hyperglycemia leads to the formation of these glycated LDL particles. Also, LDL in the environment of oxidative stress and inflammation leads to the formation of these oxidized LDL particles. And the truth is, the story of these oxidized LDL particles is very complicating. There are many factors. For example, glycated LDL particles are more likely to become oxidized. Also, other factors like metals and toxins, certain types of lipids, and our genes, these are all factors that contribute to the formation of these oxidized LDL particles. However, it's important to realize that native LDL is not such a problem. However, once we form these glycated, and especially once we form these oxidized LDL particles, they can become very problematic. So how does this happen? So taking a closer look at our blood vessels, first what happens is hyperglycemia in these advanced glycated end products, hypertension, smoking, and excessive homocysteine damages our endothelial cells. This allows the LDL particles to cross and enter past the intima in our blood vessels. Also what happens is hyperglycemia cross-links these collagen molecules. So these collagen molecules become glycated, which cross-links them, and this traps these LDL particles. So now we have these LDL particles hanging out in past the intima in our blood vessels. So there, these immune cells run into and uptake these LDL particles. And what's really important to realize is these modified LDL particles, like oxidized LDL, have unregulated uptake by these immune cells. So once these immune cells in an unregulated fashion take up these LDL particles, that is the beginning of atherosclerosis. That forms the foam cells, which then leads to plaque. So therefore, we can see that glycated and especially oxidized LDL is a big problem. And these are the factors that lead to the formation of oxidized LDL, which is essentially the byproducts of inflammation. However, there are some pretty interesting studies that show certain antioxidants, particularly vitamin E, but also vitamin C and vitamin A and other antioxidants like lycopene, these inhibit the formation of oxidized LDL. Because again, it's not this native LDL that leads to this process of atherosclerosis, it's primary oxidized LDL. So this is the beginning of atherosclerosis. And also, once these immune cells take up these modified LDL particles, that also triggers more inflammation in the arterial wall. And this is a key marker for atherosclerosis. So we also mentioned how insulin resistance can increase the levels of LDL. So how exactly does this happen? So we already explained how hypertrophic insulin-resistant adipose tissue leads to uncontrolled lipolysis and the release of these free fatty acids. So these free fatty acids are going to enter other tissues. One particular tissue they enter is the liver and hepatocytes. So once the liver has excess triglycerides, this is one of the factors that contributes to the excess formation of these VLDL particles. So all these VLDL particles, they're going to react with HDL particles. And essentially what happens is these VLDL particles take away cholesterol from these HDL particles. So once we have these cholesterol-poor HDL particles, they're more prone to be cleared out by the kidneys. So this is going to lead to low levels of HDL. So now these VLDL particles are going to react with tissues that express this lipoprotein lipase. And essentially what happens is these VLDL particles react with these lipoprotein lipase enzymes, releasing free fatty acids. So one of the tissues that express this lipoprotein lipase are adipocytes. So essentially these VLDL particles are going to release these free fatty acids to adipocytes, which then forms these IDL particles, which then mature into LDL particles, which then eventually get taken up back by the liver. But now something really important to realize is remember how this began. These hypertrophic adipocytes released free fatty acids for the liver. Now once the liver had access to all these free fatty acids and had high levels of triglycerides, this, among other factors like hyperinsulinemia and hepatic insulin resistance, remember, then this liver tissue release these VLDL particles, which then eventually give the free fatty acids back to the adipocytes. So this activates a toxic cycle where the adipocytes give free fatty acids and excess fat to the liver, and then the liver gives back that excess fat to the adipocytes. So then we have this constant cycle, this toxic cycle. And also, something important to realize is this was a bit of a simplification, and this was really just focusing on the big picture. But these VLDL particles, again, they give free fatty acids to any tissues that have lipoprotein lipase. And one of those tissues is skeletal muscle. So during this toxic cycle, they're also going to give some of these excess free fatty acids to skeletal muscle, and this contributes to the formation of this intramyocellular lipid, which again, we explained leads to skeletal muscle insulin resistance. But the point is we have this toxic cycle where these adipocytes are trying to give the excess energy, this excess free fatty acids to the liver, and the liver is trying to give this excess energy, these excess free fatty acids to the adipocytes, which then gives it back to the liver, and etc. So during this toxic cycle, some of that excess of energy, that excess free fatty acids are also going to be disposed onto other tissues like skeletal muscle. But because we have this excess toxic cycle, we're going to have increased levels of LDL. Because again, we're constantly exchanging these free fatty acids, so we're constantly activating this cycle, so we're constantly going to have higher levels of these lipoprotein particles. So this is going to lead to certain biomarkers. For example, excess triglycerides, VLDL, and LDL. Because again, this cycle is constantly activated. And again, that's also going to lead to low HDL, which again, we explain why that happens because high VLDL essentially reacts with the HDL and leads to the HDL being exposed of and cleared out by the kidneys. So all of these biomarkers are really just a sign of insulin resistance. Also, what's important to realize is these individual biomarkers really aren't that useful. And in fact, it's these ratios that are much more powerful. For example, the ratio of triglycerides to HDL, the ratio of cholesterol to HDL, and the ratio of ApoB to ApoA, which is essentially the ratio of LDL to HDL. And in fact, it's these ratios that are a much stronger predictor of long-term outcomes. For example, this ratio of ApoB to ApoA, the ratio of LDL to HDL, is an extremely strong predictor of long-term cardiovascular outcomes. Also, it's good to look for certain biomarkers of endothelial damage and inflammation. So these are some of the key biomarkers for endothelial damage, and these are some of the key markers of inflammation. So normally these LDL particles would run into the LDL receptor, which will then lead to the LDL particles to be taken up back into the liver. However, what's important to realize is these LDL particles in an environment of hyperglycemia and inflammation leads these LDL particles to become chemically modified into glycated LDL and oxidized LDL. So these chemically modified LDL particles are not as well recognized by the LDL receptor. So therefore, it takes a longer time for these chemically modified LDL particles to be taken up by the LDL receptor into the liver. So therefore, these LDL particles stay in the bloodstream for a longer period of time. They have a higher residence time. And in cardiology circles, this is a huge issue. This increased residence time is very pathological and is associated with cardiovascular disease. So increasing residence time, aka increasing the amount of time these lipoprotein particles stay in the bloodstream, increases cardiovascular disease. And it's known that insulin resistance leads to downregulation of these lipoprotein lipase enzymes, which increases the residence time of these lipoproteins. Also, high insulin and also high ingestion of fructose increases the expression of this ApoC3 enzyme on the lipoprotein particles, which also increases the residence time of these lipoprotein particles, which is associated with cardiovascular disease. Also, genetics plays a huge role in residence time. For example, there are certain genes that lead to the upregulation of these LDL receptors. So these people who are blessed with genes that lead to the upregulation of these LDL receptors lead to the clearance of these LDL particles more readily, and these people are practically immune from cardiovascular disease. Fortunately, we have beautiful but expensive drugs that increase the LDL receptors, which then decreases residence time. So therefore, these LDL particles are in the bloodstream for shorter periods of time, and therefore people live longer, and it's associated with better cardiovascular outcomes. And if you think about it, this makes sense. If these LDL particles have a higher residence time, it's more likely that they're going to become glycated and oxidized. If they're in this hyperglycemia inflammatory environment for longer periods of time, they're going to become more glycated and oxidized, which leads to this pathological cycle of forming these chemically modified LDL particles, which have high residence time, and therefore it's a more likely chance of them to bump into the arterial wall and form plaque in the first place. So therefore, increased residence time is associated with cardiovascular disease. Also, it's known that there's two types of phenotypes when it comes to LDL particles. There's phenotype A, where we have these large, fluffy, buoyant LDL particles, and there's phenotype B, where we have small, dense LDL particles. And you want to avoid having phenotype B, and you want to have phenotype A with large, fluffy, buoyant LDL particles. And there have been certain experiments that have shown that a diet with high refined carbohydrates lead to phenotype B, where you have these small, dense LDL particles. And these are pathological because these small, dense LDL particles are not as well recognized by the LDL receptor. So therefore, they have a higher residence time. They're in the bloodstream for longer periods of time, which again can run into environmental insults like inflammation, glycation, which then we know can lead to oxidized LDL, which we know is pathological. And again, a high residence time means there's a more likely chance for these LDL particles to bump into the arterial wall and form atherosclerosis. And also, it's hypothesized that the smaller LDL diameter makes it more easily to sneak past the endothelial wall and therefore form the atherosclerosis in the first place. So again, there are certain studies that show for certain populations that it's a high carbohydrate diet that leads to the formation of these small, dense LDL particles. And the truth is it's complicating, and there are a lot of different factors and theories for the causes of these LDL particles, but a high diet in refined carbohydrates for certain populations have been shown to lead to small, dense LDL formation. And it's known that hyperglycemia can be pathological for many different reasons. For example, excess glucose can be taken up by cells and converted into sorbitol in an unregulated fashion. And this sorbitol is osmotically active, which can lead to the lysis of certain tissues. And also, this process of forming sorbitol uses up NADPH, which uses up this reduced glutathione, which is our endogenous antioxidant. So this process of glucose turning into sorbitol is sopping up our endogenous antioxidant. Also, it's known that high glucose inhibits the immune system through various mechanisms. And if you're interested, these are some of the mechanisms. But what's interesting is it's various mechanisms in which high glucose inhibits the immune system. And also, through some complicated biochemistry, high glucose also leads to microvascular damage, the damage of very small blood vessels. Also, sometimes people focus too much on hyperglycemia and forget about the pathological effects of hyperinsulinemia, this excess insulin. For example, high insulin leads to hypertension through various mechanisms, which is really interesting. Usually people don't think of insulin raising blood pressure, but it increases renal sodium absorption. It activates the sodium-potassium transporter, which increases blood sodium, and it also activates the sympathetic nervous system, leading to vasoconstriction, which raises blood pressure. And the way you should think about it is if you have excess sodium, the kidneys get rid of that sodium and therefore lowers blood pressure. However, insulin blocks the kidney's ability to remove that sodium, so that sodium stays in the bloodstream and leads to hypertension. However, there are certain things you can do to help your kidney get rid of that sodium. For example, a diet high in potassium and magnesium helps the kidney get rid of that sodium. And also, there are very interesting clinical studies on humans that show if you eat a diet high in sodium, that will increase blood pressure. And then it'll take a couple hours for the kidneys to get rid of the sodium to lower the blood pressure. However, if you eat a diet high in sodium and you eat that high sodium diet with excess water consumption, that decreases blood pressure. So that's a sign that it's the osmolarity of sodium in the blood that helps the body regulate blood pressure. So those are very interesting studies that show if you eat a lot of sodium, if you eat it with potassium, magnesium, and water, that can help the kidney get rid of that sodium and therefore lower the blood pressure. And remember, we explained if you eat excess fructose, it's taken up by the liver. Then the liver oxidizes that fructose in an unregulated fashion, essentially overwhelming the mitochondria, which then leads to excess formation of these end substrate products, which then lead to the formation of excess acyl-CoA and this lipid droplet, which then leads to hepatic insulin resistance. But not just that, when this fructose is metabolized, it uses up ATP, which forms ADP, which then forms AMP, which then gets catabolized and forms uric acid. So this uric acid is also associated with certain markers of metabolic syndrome. But something important to realize is that this uric acid essentially inhibits the formation of nitric oxide, which also increases blood pressure. So the way you should think about it is our body releases nitric oxide, which opens up the blood vessels and lowers blood pressure. And that's one way our bodies regulate blood pressure. However, excess fructose leads to the formation of uric acid, which inhibits the body's ability to regulate vasodilation, which then increases blood pressure. So this hypertension is extremely important because this hypertension essentially damages the endothelium, which then allows LDL particles to sneak in and lead to the formation of plaque and atherosclerosis. Also, it's important to realize that our endothelium has this important protective glycocalyx layer. And this glycocalyx layer essentially protects the endothelium and prevents LDL particles from crossing into the endothelium and causing atherosclerosis. However, there have been studies that show that once you eat a meal in carbohydrates, it temporarily destroys the glycocalyx. So if you're constantly eating carbohydrates, you're constantly destroying the glycocalyx, allowing LDL particles to cross and enter and lead to atherosclerosis. And also, it's important to realize that homocysteine is a huge factor that leads to endothelial damage. And this is an extremely important factor, and just because I didn't have enough time to go into more details doesn't mean that this isn't one of the most important factors in atherosclerosis. So what can we do about insulin resistance? Well, first of all, we have these incredible drugs which are literally gifts from God. For example, metformin activates AMP kinase, which has been shown to lead to incredible long-term outcomes. Also, we have these SGL2 inhibitors, which essentially tell the kidney to pee out all that excess glucose, which was causing all that trouble. And there are other drugs, and the truth is, scientists have gone into great details learning the biochemical mechanisms of diabetes, and they've created these incredible drugs that target some of those mechanisms. Also, if you want to stay a little bit on the safer side, you can also, I very much like cinnamon. Cinnamon has been shown to reduce blood glucose levels by up to 1/4. I also like curcumin. Curcumin downregulates NF-kappa beta, which is the key master regulator of inflammation. So curcumin can lower systemic inflammation. I also like chromium and magnesium because they essentially help lower blood glucose levels and help your cells become more sensitive to insulin. I like berberine because it upregulates that LDL receptor, lowering residence time. And I also like vitamin E and lycopene because they're antioxidants and lower the amounts of oxidized LDL. So these are a couple of safer natural supplements that have been shown to have real biochemical effects. Also, there isn't evidence to prove this. However, theoretically, if you're constantly eating glucose and living a sedentary lifestyle where you're never fasting and you're constantly consuming glucose and carbohydrates, you're going to constantly burn glucose as a source of energy. You're going to have high glycogen stores because you're constantly eating glucose and you're not exercising and using up that glycogen. So therefore, you're going to have high glycogen stores and you're constantly going to be burning glucose as a source of energy to create ATP. So therefore, because you're constantly using glucose as a source of energy, all that excess energy from your diet is going to be stored as fat. So now you're going to have excess storages of fat, which we know cause problems and lead to insulin resistance and systemic inflammation. However, what happens if you become active and you start restricting carbohydrates and you start fasting? Well, if you're more active and fasting and restricting carbohydrate intake, you're going to use up all your glycogen. You're going to use up all your glycogen and burn all that glucose. So once you burn all your stored carbohydrates, then and only then will you start burning your fat. And once you start burning your fat, then you'll have lower levels of fat. And remember, that fat was causing inflammation and insulin resistance. So again, if you're active and restricting carbohydrates and fasting, you're going to burn that fat. So now you have less of that fat, which was causing all that trouble. And also, if you're active and exercising, that leads to mitochondrial biogenesis where you have extra mitochondria. So therefore, you're not overwhelming the mitochondria. You have excess mitochondria, so you're able to burn that fat and other excess calories. So the key idea are there are two different lifestyles. There's one lifestyle where you're sedentary and you're constantly all day long eating carbohydrates. So in that circumstance, you're going to have a lot of glycogen, so you're constantly going to burn glucose as your source of energy. So all that excess fat is just going to stay around and become sedentary lipid droplets, which we know causes problems. However, if you're active and you go through periods of fasting and carbohydrate restriction, you're going to use up all that glycogen. So you're not going to have stored carbohydrates as a source of energy. So then you're going to start burning fat. And then you're going to get rid of that fat because you're going to burn that fat, so you're going to have less inflammatory fat lying around. So again, this is just a pet theory of mine. But if you think about it, most of our evolution, we were dealing with starvation. So the genes that were selected for were the genes that helped us during starvation. But now in our modern age, we're flooded with calories. And what happens is we eat excess calories in the form of branched-chain amino acids and fructose, and for certain people, alcohol. So what happens is we didn't evolve to have the genes and enzymes that regulate the oxidation of fructose, branched-chain amino acids, and alcohol. So essentially what happens is our cells take up branched-chain amino acids, fructose, and alcohol. And again, we didn't evolve to have the genes that regulate the oxidation of these specific biomolecules. So therefore, our cells will oxidize these biomolecules in an unregulated fashion. And essentially, they're going to create substrates that overwhelm the mitochondria. So when you overwhelm the mitochondria, because again, you're oxidizing these biomolecules in an unregulated fashion, you're going to create too much substrate, overwhelming the mitochondria, which then leads to the formation of lipids, which again, we already explained, causes problems. But again, if you're exercising in certain tissues, you're going to have excess mitochondria, which then if you have excess mitochondria, then you're not going to be able to overwhelm the mitochondria and you'll be able to burn all the substrates and therefore theoretically have less ectopic fat. However, what is not a theory and has been proven lately with some very high quality studies is that it's this hepatic fat, which is one of the best predictors of metabolic syndrome, not subcutaneous fat, not visceral fat. In fact, it's this hepatic fat, which is one of the best predictors. And essentially what happens is when you have fatty liver, when you have excess lipid droplets in your liver, that leads to hepatic insulin resistance. And when you have hepatic insulin resistance, that leads to uncontrolled gluconeogenesis, and it also leads to hyperinsulinemia. And through complex mechanisms also leads to high VLDL output. So therefore, it's been shown that this fatty liver is one of the best predictors of long-term serious unhealthy outcomes for cardiovascular and cardiometabolic disease. Also, there was this beautiful study that showed 10 days with no fructose, and that was the only difference. It was an isocaloric diet. So 10 days with no fructose led to a 22% reduction in liver fat. So essentially cleaned out liver fat, and a 7% reduction in visceral fat, and a 50% reduction de novo lipogenesis. So 10 days with no fructose cleaned out the liver and strongly improved insulin resistance. For example, we can see this oral glucose tolerance test, which is the gold standard of insulin resistance. So this green represents the oral glucose tolerance test with normal fructose consumption. And this blue represents 10 days with no fructose, the oral glucose tolerance test, which was much better, which shows better insulin sensitivity. And this shows the levels of insulin. So again, the green is normal fructose diet. Then the blue is 10 days with no fructose. So the idea is that 10 days with no fructose reduced the levels of fatty liver, which then strongly improved insulin sensitivity. And some people believe that insulin resistance actually begins with the liver. It's this fatty liver that leads to hepatic insulin resistance that leads to a lot of these markers of metabolic syndrome. The truth is, no one knows for sure, but maybe it's possible that there are very fortunate metabolic dynamics where it takes years to develop fatty liver. But due to the energy turnover dynamics, given the right physiological circumstances, this fat in the liver is the first to go in these right physiological circumstances, which can theoretically reverse a lot of the symptoms of metabolic syndrome and restore insulin sensitivity. So I'm not saying this to be self-deprecating, but the honest objective truth, that is, in 5 years, we're going to find that 50% of what I said in this video is wrong or incomplete. And even now, I admit that 100% of what I've said is a huge, gross oversimplification. And the reductionist explanation of disease is always the wrong explanation when it comes to biology, because biology is complex and messy and irreducibly complex. But the purpose of this video was just to share some of the current theories and ideas being thrown around in diabetes research. And really, the hope is that this is a starting point for your own investigative research on these diseases.